Hello and welcome back. Today we're going to be talking about lung capacities and volumes. The lungs are a very important part of the respiratory system, being able to get oxygen into the body to get it to the cells and perfuse to all the liquids and cells so they can have all of their functions is a very important part of anatomy. My name is Matthew Greer. This was prepared for Grand Canyon University Biology 520, which is Concepts of Human Physiology 2. My instructor is Dr. Sandra Barrera, and this was prepared on the 2nd of February in 2022. So let's jump right in. All right, now let's talk about how lung volumes are determined. The device that is used is called a spirometer, and it looks something like this. It has a flexible tubing. It has a mouthpiece. There's an indicator here. There's these kind of volume gauges here. There's this little marker here and a piston. What happens is as an individual is going to blow through this mouthpiece, this will raise this up, and as that raises up, this indicator will either fall down or go up to the top here, depending on if the individual is blowing too hard or not hard enough into this mouthpiece. So the procedure will be the individual will take a deep breath in, all the way in, put the mouthpiece in, and then blow all the air out of their lungs until they're done. And then this little marker here is used to determine how high that went, so that when this piston returns back down to the bottom, they have good data on that. So the graph, the spirogram, can look a little bit different. We're going to look at tidal volumes coming up next, but this is one where we have a big inspiration, and then they're holding it at the top. When we look at tidal volumes on the next one, we're going to have both up and downs. This is going to be inspirations are going to be ups, and expirations are going to be downs. All right, next we're going to talk about lung volumes and capacities. And the first one we're going to talk about is the tidal volume, or VT. This is the volume air entering or leaving the lungs during a normal breath. So inspiration going up, expiration going down. In a normal breath, this is the volume of air that's going in and out. Just a very small part of this graph. Now our next one is the inspiratory reserve volume. And it's this chunk here on the graph. And this is the extra air that you could inspire outside of the tidal volume, so outside of this normal range here, anything above this. And to do this, we have to maximally stretch the diaphragm, we have to stretch the external intercostal muscles and the accessory muscles to get this extra volume inside. All right, our next one is the inspiratory capacity, and this is the maximum volume of air that can be inspired at the end of a quiet, normal expiration. So we have normal tidal volume. As we breathe all the way in, this is the amount of air that we can get in. So to get this, we take this IRV here plus the VT, the tidal volume, and that's how you get your inspiratory capacity. Our next one is ERV, the expiratory reserve volume. And it is this extra volume of air here that can be expired by contracting the expiratory muscles. So it's unlike our other one where we were inhaling and getting extra, this is the extra that we can push out during a normal, after a normal breathing kind of process. So it's normal here, the extra air that gets pushed out there. The residual volume, this little green guy down here, is the minimum volume of air that is present even after this ERV, this maximum expiration here. We can't determine what the RV is with a spirometer. Spirometers are much better at inspiration, but there is a fraction of air that is always inside the lungs that you can't always breathe out. And on a normal breath, normal tidal volume here, we aren't breathing out a lot of air here. There's a lot of air that's staying inside the lungs that's getting recycled as we breathe in our next breath. All right, the FRC, the functional residual capacity. This is at the end of a normal tidal volume, just normal breathing here. This is the amount of air that's left in the lungs. It is quite a significant amount of air that's left in the lungs. before the next breath. Vital capacity, this is this chunk here. This is the maximum amount that can be moved in a single breath. So the most you can get if you exhaled all the way and then inhaled all the way and exhaled all the way and inhaled all the way. This is your vital capacity. We don't usually breathe using vital capacity because it's exhausting to do so. There's a lot more muscle movement. The lungs, remember, are built to utilize pressures and atmospheric pressure to kind of be more of a passive system. That's why this tidal volume is a much smaller chunk here versus the vital capacity, which is this huge kind of wave here, much larger, and there's a lot more muscles being used, so it's a lot more exhaustive process. And then the TLC, the total lung capacity, this is the total amount of air that can be held in the lungs at any time. So this is a addition of the FRC and the IC. You can look at it as inspiratory capacity and the functional residual capacity. If you add those two together, you can think of it as all these small parts added up together. But this is at any point in time the total amount of the total amount of air, that can be present inside of the lungs. All right, let's talk now about the effects of smoking and stopping smokers from smoking by showing them their lung age. And this lung age is also kind of determined by spirometry. So this study from 2008 encouraged participants to stop smoking. Spirometry can detect an increase in obstructive lung damage after about 20 years of smoking, which typically presents at age 35 in smokers. It can present earlier if they're smoking more cigarettes a day. It can present later if they're smoking less cigarettes a day. This study uses lung age, which is a concept that was developed in 1985, and it shows the spirometry data of age in years on the bottom here, and then the amount of lung being used here on the left-hand side. And this red on the top is people who have never smoked at all, and it shows their lung capacity over time. And as they get older, it starts to drop off, but not nearly as fast as this blue line. And this blue line is individuals who are susceptible smokers. So if you are a smoker or you know a smoker, they're probably more on this line here. Now, this study is involving trying to get people to stop. So there's some extra lines on here. This green one here that's dotted shows this is what the lung capacity, the spirometry data would look like if they stopped smoking at 45. And this one here is what they would stop smoking at 65. So this table uses another kind of lung capacity term, which is the forced expiratory volume in 1 second, or the FEV1, which is the volume of air that can be exhaled in the first second of expiration when we determine vital capacity, so that total amount there. And usually, this number will be around 80% of the vital capacity of the lungs. So in this study, to get the data and to set kind of a baseline, individuals were asked a questionnaire where they were looking at things like antibiotics and steroids for respiratory infections, whether or not the patients had emphysema, asthma, diabetes, blood pressure treatments, stroke, or a history of coronary heart disease. And they made this baseline to see where these individuals should be. They then all had spirometer readings taken, and then were told that in 12 months they were going to get another spirometry reading, and they were all encouraged to stop smoking and given access to some information on smoking cessation clinics. Now, an important thing in this study is each participant had to have 2 spirometry readings that were within 5% of each other, 5% kind of percentage there on the lung capacity, lung function. And this was to determine that we had good, solid numbers per baseline. All the patients in this study, all the participants had the effects of smoking on the lungs explained, and they were told that stopping smoking wouldn't stop this kind of progression of aging on the lungs, but it would slow it down. Now, not every individual is shown this awesome lung age chart, and in fact, the control group wasn't shown this data at all. So then, a little bit more on the study itself. After 12 months, the individuals would come back. They were tested for cotinine, which is produced when nicotine is broken down in the body. The half-life of this chemical is 20 hours. So this testing for it to see if it's been metabolized in the last couple days is pretty accurate, up to 24 to 48 hours. So what this tells us is, of the individuals who came back in, the ones that should have quit won't have any of the substance in their bloodstream. The control group got their results from their spirometry, their initial spirometry, as their FEV1, their liters per second, with no other information about lung age or effects like that. They were given the information on the cessation and how to, reach those clinics, but none of the lung age information was dispersed to them. The experimental group was told their lung age and shown the graph and explained on the graph what it means for if they are a 50-year-old individual and they're a smoker. If they stop now, they could slow down that aging versus keeping going down that same blue line there. So there's 561 participants in this. And the results came back, and it suggested that using this lung age to motivate individuals is an effective method. The individuals that came back that had seen the graph and thought about their lung age were statistically significantly more likely to quit smoking, and that group had a lower daily cigarette usage than the control group that didn't get any of this information. The researchers in this study kind of postulated that a lot of the individuals were younger, and so they seen that there was still time for them to stop smoking and kind of get back on this normal lung age kind of chart here instead of starting to go down this blue line. So they think that that was a really big motivation for these patients to stop. For this presentation, I used the following references. This one on lung age is by Parks, Greenhalgh, Griffin, and Dent. It's a really good one, using spirometry to kind of test these. There's a lot more information on that paper about different, history and other tests that were ran. And then for the physiological portion of this presentation, I used the textbook Sherwood, Human Physiology from Cells to Systems. It's got a lot of good things about spirometry. It talks about restrictive and obstructive lung disease in spirometry, but I didn't have time to get into that in this presentation. I hope you learned something about the lungs. I hope you found the study on the lung age interesting as I did. And thank you for watching. Have a great rest of your day.